In drug development, millions of compounds begin the journey toward regulatory approval, but the reality is a funnel of attrition. Only a fraction survive the initial screening, and ultimately, just a single candidate emerges for human use. Yet, even among the candidates that make it to human clinical trials, over 90% fail. They fall short because of unexpected toxicity or a complete lack of efficacy in human patients. What makes this failure rate striking is it happens after candidates have cleared the mandated gold standard of safety testing, in vivo mammalian models. For decades, toxicology has relied on the assumption that a complex biological response in a rodent or a rabbit will reliably mirror outcomes in humans. We are using mammalian approximations to predict highly specific human physiology. This 90% failure rate indicates a systemic mechanical bottleneck. The reliance on legacy animal models produces a high volume of misleading data, necessitating a move towards testing focused on human molecular events. The root of these clinical failures lies in evolutionary divergence. Different species possess entirely distinct physiological and immunological pathways. Take brain development, for example. Human neural progenitor cells behave differently than those found in rodents or non-human primates. Viruses like Zika target human neural pathways that simply do not exist in macaques. The same divergence applies to immune responses. The pyrogenic thresholds, the exact point at which a body triggers a fever, vary across species. A rabbit's inflammatory response does not reliably mirror a human's. Because human pathophysiology is uniquely human, accurate prediction depends on models built on human biology. When evolutionary proxies fail to account for these specific differences, the resulting data cannot reliably guide clinical outcomes. Recognizing this translational gap, regulators are changing their framework. The traditional 3 Rs, replacement, reduction, and refinement, are being superseded by a formalized mandate for new approach methodologies, or NAMs. Global regulators are writing rigid phase-out schedules using a targeted basket system. Basket 1 mandates immediate decommissioning of legacy tests like the rabbit pyrogen test. Baskets 2 and 3 require strict reductions in primate use by 2030, tracking toward a total phase-out by 2035. Regulators are actively removing legacy in vivo assays from their requirements and demanding protocols built entirely on mechanistic human data. The first step in this transition was recognizing the physical limitations of early in vitro models. Traditional two-dimensional cell cultures lack spatial architecture and fail to replicate the complex extracellular matrix interactions that define living tissue. Modern bioengineering solves this by growing stem cells in 3 dimensions. Suspended in fluid, these clusters rapidly divide and self-assemble into complex, layered tissue formations. These are 3D organoids. Because they are derived from human stem cells, they self-organize to mimic human organ-level architecture. This structural fidelity enables researchers to accurately model complex viral infections like Zika directly on human neural tissue. To push the technology further, researchers utilize microphysiological systems, commonly known as organs on a chip, to replicate the physical and mechanical environment of the human body. These systems contain tiny microfluidic channels lined with living human cells. By pumping blood substitutes or nutrients through these channels, the chips simulate critical mechanical forces, like the fluid shear stress inside blood vessels or the cyclical strain in expanding lungs. By linking multiple organ-specific chips together, scientists create body-on-a-chip networks. This setup allows researchers to track systemic drug metabolism, observing how a compound processed by a liver chip triggers cascading toxicities in a connected heart chip. Simulating interconnected multi-organ fluid dynamics in vitro directly maps systemic toxicity. It eliminates the historical defense that only a live, intact animal can demonstrate whole-body effects. One of the most rigorous replacements happening right now targets the TRACE rabbit eye irritation test. The TRACE is a subjective assay that relies on visual scoring of conscious animals. It is currently being replaced by the HETCAM assay. The biological baseline for the HETCAM test begins here, an intact, fertilized white leghorn egg, incubated for exactly 10 days at carefully controlled humidity. The precise mechanical preparation involves carefully removing the shell over the marked airspace. The inner white membrane is moistened with saline and stripped away to expose the chorioallantoic membrane, or CAM. The biological advantage of this membrane is that it provides a complex, highly vascularized respiratory network. Most importantly, at 10 days of incubation, the CAM completely lacks functional nerve fibers and pain receptors. Researchers apply exactly 0.3 milliliters of a test substance directly onto the CAM. They then observe the network for 3 specific, measurable reactions: hemorrhage, where blood leaks from the vessels; lysis, where the vessel walls dissolve; and coagulation, marked by cloudy spots of denatured protein. The assay operates under a strict temporal constraint. Using a chronometer, researchers meticulously record the exact onset time of each vascular reaction within a tight 300-second window. These timed onset values do not rely on human judgment. Instead, the raw temporal data is fed into an algorithmic computer program that calculates a precise, objective irritation score. By isolating the vascular mechanics, highly controlled biomembrane assays generate tightly reproducible toxicological data. It is a mathematical output, free from the interpretive variables of scoring a distressed, conscious mammal. A similar transition has already rendered the rabbit pyrogen test obsolete. For decades, this was the standard method for detecting fever-inducing contaminants, like bacterial endotoxins, in injectable medicines. But the test has scientific vulnerabilities. Restrained rabbits possess different pyrogenic thresholds than humans, and their body temperature frequently spikes simply from the stress of being handled in a laboratory. The biologically superior alternative is the monocyte activation test, or MAT. Its accuracy and reproducibility directly triggered the European Pharmacopoeia to ban the rabbit test outright. The MAT mechanism is entirely in vitro. It utilizes donated human blood or specific human cell lines. When exposed to pyrogens, these cells react by releasing human cytokines, such as interleukin-6, mimicking the human immune system. Triggering a direct human cytokine response provides a safety threshold based on human immunity, a level of precision that cross-species extrapolation reach. Beyond physical biological assays, the architecture of replacement is expanding into computational intelligence. The primary tool here is the quantitative structure-activity relationship model, QSAR. QSAR models use mathematics to scan a novel molecular structure against databases containing tens of thousands of known compounds. The algorithm rapidly isolates dangerous toxicophores, structural fragments historically proven to cause specific cellular damage. Next, researchers employ physiologically based pharmacokinetic modeling. This process mathematically simulates the entire life cycle of a substance inside the human body, tracking its absorption, distribution, metabolism, and excretion in real time across digital organs. The comparative data is definitive. Advanced AI models now achieve an 87% accuracy rate in predicting chemical toxicity. By contrast, the reproducibility of the traditional animal tests for the exact same compounds sits at 81%. Algorithmic analysis mapped strictly to human biological parameters mathematically eclipses the unpredictable variables of in vivo mammalian testing. The transition away from animal testing is driven just as aggressively by the unsustainably high costs and slow operational pace of legacy research and development. This data table compares assay costs. Looking at phototoxicity testing, a standard mammalian assay costs upwards of $11,500. Running the direct in vitro equivalent drops that cost to just $1,300. The efficiency scales across endpoints. Replacing the rabbit pyrogen test with the monocyte activation test generates a 90% financial savings per batch. This creates a fail-fast financial philosophy. By identifying non-viable therapies early in development through human-relevant data, companies prevent burning hundreds of millions of dollars on doomed late-stage clinical trials. Yet, the widespread adoption of these methods faces a severe structural barrier. It is a documented phenomenon known as animal methods bias, and it is deeply ingrained within the scientific publishing and grant funding ecosystem. Institutional conservatism often demands that researchers include in vivo data to validate a study, even if the in vitro data is more accurate. This forces scientists into anticipatory testing, conducting irrelevant animal experiments solely to appease biased grant committees and peer reviewers. The continued use of legacy animal models is sustained by institutional habit rather than empirical predictive validity. Overcoming this bias requires a change in how regulatory science is organized, moving away from the goal of one-to-one replacement and toward the framework of placement. Placement means researchers stop validating precise new technologies against historically flawed animal data. Instead, they validate biological mechanisms directly against human adverse outcome pathways. This mechanistic validation forms the foundation of the weight of evidence approach. This flowchart illustrates how modern pharmacology synthesizes risk without relying on a single mammalian proxy. The weight of evidence framework aggregates datasets for AI computational simulations, microfluidic chips, and precise in vitro assays. These streams converge into a single, highly predictive human risk profile. Predictive toxicology has crossed a threshold. We are now in an era where integrated, human-centric methodologies have rendered whole animal testing a scientifically obsolete metric.